So good afternoon. I am Maria Isabel, a PhD student from Cádiz University, and I am going to talk about the effect of the synthesis temperature and palladium doping of cesium lead tribromine quantum dots for biomedical applications. Firstly, I'm going to talk about the luminescent nanofluid for biomedical applications. Next, about one of the materials used for these applications, that is perovskite. In particular, I am going to comment its structure, the success of perovskite in this field, and the synthesis method used to obtain the perovskite quantum dots. Then I'm going to comment the study of the synthesis temperature and palladium doping of cesium lead tribromine quantum dots that I have done in our lab. So I will comment the site analysis, the photoluminescence spectroscopy, the time-resolved photoluminescence spectroscopy, and the ultraviolet visible spectroscopy, and finally the conclusions about those results. So currently, the development of luminescent nanothermometry has allowed to precisely register the temperature in real time, which has many biomedical applications such as diagnosis and medical treatment. The luminescent nanothermometry uses luminescent nanoparticles, which show significant temperature-dependent photoluminescent properties. The main requirement to use this technique for in vivo applications is having an excitation and emission wavelengths that fall in any of the three biological windows, ranging the first biological window between 650 and 950 nanometers, the second biological window between 1000 and 1350 nanometers, and the third biological window between 1550 and 1870 nanometers. This requirement is key to get that the absorption and the scattering of the light by the biological tissues are minimal. Those luminescent nanofluids find application mainly in two fields: biology, where they are used as biomarkers, and physics, where they are used, for instance, as nanoscale thermal sensors. But how do the luminescent nanothermometers work? They work detecting changes in any of the photoluminescent properties of the materials, which are associated to temperature variations such as the intensity of the emission band, the position, the emission radio, the shape of the band, or the lifetime. The most widely extended method to determine the temperature is the radiometric method, which determines the temperature by measuring the intensity radio between two emission bands. With this aim, the luminescent intensity radio expression is used to get the temperature value, and the relative sensitivity value gives an idea about which nanothermometer is better. The nanothermometers are usually made of semiconductor quantum dots and Pd-doped dielectric nanoparticles. Both have their advantages and disadvantages. In the case of semiconductor quantum dots, their sensitivity is high, but to get the temperature value, one single band is measured. On the contrary, Pd-doped dielectric nanoparticles have a low sensitivity, but the temperature is determined by measuring the intensity radio between two emission bands. Hence, we decided to prepare luminescent nanofluid made of inorganic halide perovskite quantum dots, as their sensitivity is higher, and we dope them in V position to tune their photoluminescence in order that they emit in the first biological window and can be applied in nanothermometry. Perovskite are materials described by the formula ABX3, in which A is a monovalent or organic cation, such as formamidinium or methylammonium. B is a divalent inorganic cation, such as lead or palladium. And X is a halogen, such as chlorine, bromine, or iodine. There are two important factors to study the possibility that a perovskite structure is obtained: the tolerance factor, which is calculated to study the formability of halide perovskite, and it is defined as shown in the screen, and the octahedral factor, which is the ratio of the big cation radia and the halogen radia. It assesses whether the B-side cation can fit within the octahedral hole of the X-side anion sublattice. Perovskite nanomaterials have recently emerged as promising nanomaterials due to their excellent optoelectronic properties, such as high photoluminescent quantum yield, narrow bandwidth, or their tunable photoluminescence by doping on A, B, or X position. What stabilizes the crystalline structure modifies the photoluminescent properties or even improves the optoelectronic efficiency. Thus, those amazing properties have drawn considerable attention for their application in diverse devices, including light-emitting diodes, laser, photodetectors, sensors, solar cells, or luminescent nanothermometers. Nevertheless, there are still some challenges in this field, like the blue emitters' low efficiency, the poor stability of those compounds, or the fact that they don't emit in the biological window for their application in luminescence nanothermometers. Nevertheless, as I will comment later on, in our study, we took advantage of the fact that we can tune their photoluminescent properties by doping them, and we overcame this problem. The hot injection method was employed to obtain perovskite quantum dots, where A is cesium, B is lead or palladium, and X is bromine. This method consists of 3 steps. The first step is the preparation of cesium oleic. For that, cesium carbonate was loaded into 3 net flasks along with octadecene and oleic acid. Dry for 1 hour at 120 degrees and then heated under nitrogen to 150 degrees until all the cesium carbonate reacted with oleic acid. The second step is the synthesis of the perovskite quantum dots. For that, octadecene and lead dibromine, palladium dibromine, or their mixtures were loaded into a 3 net flask and dried under vacuum for 1 hour at 120 degrees. Dry oleilamine and dry oleic acid were injected at 120 degrees under nitrogen. After complete solubilization of the salt, the temperature was raised to 130 degrees to 190 degrees for tuning the nanocrystal size. And cesium oleic solution, prepared as described before, was quickly injected, and 5 seconds later, the reaction mixture was cooled by the ice water bath. The final step was the isolation and purification of quantum dots. For that, the crude solution was cooled down with water bath, and aggregated nanocrystals were separated by centrifuging at 0 degrees and 7,000 RPM for 50 minutes. After centrifuging, the supernatant was discarded, and the particles were redispersed in toluene, forming long-term colloidally stable solutions. In the case of palladium-doped cesium lead tribromine quantum dots, it was not possible to obtain the precipitated quantum dot by centrifugation, evaporation, or by drying under vacuum. For that reason, the colloidal suspension of perovskite quantum dots was kept in octadecene. As it was necessary to check out the amount of palladium with which it was possible to carry out the doping of the cesium lead tribromine, keeping the perovskite structure, we synthesized perovskite bulks with the composition shown in the screen, where X is 0, 0.25, 0.50, 0.75, or 1. This was done by dissolving 2.48 multiplied by 10 raised to the minus of 3 mole of cesium bromine, lead dibromine, and palladium dibromine in 2 milliliters of dimetal sulfide in an ultrasound bath, and then the mixture is heated in a hot plate at 60 degrees up to obtaining powders of the perovskite, We found out that for composition in which X is higher than 0.50, the perovskite structure is not obtained. Thus, we decided to dope the perovskite with 0.25 palladium. And now I am going to comment the result of the characterization of the samples. In this slide, we can see the 10 images of the fission lead tribromine quantum dots obtained at 130, 150, 170, and 190 degrees using ImageJ software. We analyze the size of the nanoparticles at different temperatures, which ranges between 6.5 and 18.2 nanometers. Those results show that the size of the nanoparticles increases gradually when the temperature is increased. In the case of the palladium dope fission lead tribromine quantum dots, we can observe the temperature effect on the quantum dot side, which ranges between 6.3 and 10 nanometers. To explore the crystalline configuration of the different samples, time analysis is carried out. Bright field images were acquired, and in the cases in which well nanoparticles were obtained, we did a selected area diffraction pattern site study. In this regard, Figure 1A shows 10 bright field micrography of fission lead tribromine nanoparticles, annealed at 170 degrees. It can be observed that the nanoparticles are square-shaped, conforming a structure that is similar to a 20 nanometers edge-side square-based springs. To distinguish well-aligned nanoparticles, high-resolution transmission electron microscopy images were acquired for a variety of nanoparticles that are shown in Figure 1B. We consider that a nanoparticle is well aligned when atomic columns could be appreciated, and the fast Fourier transform shows a comprehensible pattern. Focusing on the fission lead tribromine annealed at 170 degrees, this sample is expected to form an orthorhombic lattice belonging to the PNMA space group, as you can see in the figure. So fission lead tribromine in orthorhombic configuration is expected to elongate along Z-axis or 001 crystallographic axis. Taking this into consideration, and once the camera constant, which depends on the observation condition, is determined, the spacing can be calculated as shown in the expression, where lambda per multiplied by L is the camera constant, measured in angstrom per pixel. D is the D spacing in angstrom. R is the geometric distance between the two adjacent side spots, here measured in pixels. As the geometric distance between two side spots corresponds to the D spacing between these two reciprocal lattice spots, measuring on the corresponding side in Figure 1C allows determining the interplanar distances. On the other hand, in side patterns, each diffraction spot or reciprocal lattice point represents a set of equally spaced real lattice planes. Thus, measuring the angle between the HK0 and HKL Lagrange lines represented by dashed lines in Figure 1C equals the angle between the corresponding real lattice planes. By using this data and the LM relation presented in Figure 1C, 010 pole of an orthorhombic system is determined. So the sample obtained at 170 degrees is coincident with a fission lead tribromine in orthorhombic configuration, with the lattice parameters shown in the screen, where A and B are equal and C is different from the other two parameters, which is understandable as it is an orthorhombic space group. By repeating this procedure, crystalline structure of the palladium dope samples can be determined. Concerning those samples, important structural changes are detected through the annealing process. Indeed, samples annealed at 130 degrees shows nanoparticles with a hexagonal section, which can be approached to a circle with a diameter of 20 nanometers. These nanoparticles are accompanied by other small nanoparticles with a circular section of 5 nanometers radius, shown in figure 3a. Unfortunately, we found no nanoparticle well aligned with the electron beam, so no high-resolution transmission electron microscopy or site data could be presented here. The above-mentioned case can be compared with that of palladium-doped sample annealed at 190 degrees. Figure 3b shows bright field micrography of such nanoparticles. It can be observed that the anneal results in nanoparticles with a square-based section with approximated dimensions of 5 per 5 per 50 nanometers. Our explanation for this phenomenon is that these nanoparticles could be composed by a variety of domains that induces some strain realization in the lattice. An orthorhombic configuration is detected with lattice parameters shown in the screen. These results are quite similar to that obtained in the original cesium lead tribromine samples that I commented before. Now I am going to comment the result of the photoluminescent spectroscopy characterization. In the screen, we can see the normalized photoluminescent spectra of the perovskite quantum dots obtained under excitation at 473 nanometers. In the case of cesium lead tribromine, we can see a redshift of the emission band as the temperature is increased. This same effect can be observed as the synthesis temperature increases in the case of the palladium-doped samples. If we compare the photoluminescent spectra of the doped and undoped sample at the same synthesis temperature and under the same excitation wavelengths, meaning in this case 130 degrees and 190 degrees, and under excitation with the laser 473 nanometers, we can see that the effect of palladium doping is a blue shift in the main emission band. Nevertheless, in the case of palladium-doped perovskite quantum dots, the band obtained under excitation at 473 nanometers isn't the only one which can be observed. Under excitation at 785 nanometers, we can see a band at around 875 nanometers, which was not observed in the undoped samples. Thus, the palladium-doped samples could be applied in luminescent nanothermometers, as they can be excited using a near-infrared laser, and they emit in the first biological window. We also determine the lifetime of the samples by using the time-resolved photoluminescent spectroscopy. By doing a first-order exponential decay fitting to the decay curves obtained under excitation at 467 nanometers, we obtain the lifetime which are shown in the table. Those lifetimes are low, which shows that the desactivation of the samples after being excited occurred quickly through fluorescence. In the case of palladium-doped samples, the lifetime are also short and quite similar for the different temperatures, as well as you can see in the table. To finish the characterization part, I am going to talk about the obtained ultraviolet visible spectra. As it is shown in the graph, in the case of cesium lead tribromine quantum dots, we can see one single clear absorption band, which is redshifted from 485 to 517 nanometers as the temperature increases. In regard of the doped samples, there is one single absorption band at around 407 nanometers, which is blue shifted comparatively with the ones obtained for the undoped samples. We can also see the same effect through the temperature as in the case of the undoped samples. In conclusion, those results show that in all cases, the crystalline system is orthorhombic A, the effect of the synthesis temperature is the gradually increase of the nanoparticle size, which produce a redshift on the main band obtained under excitation at 473 nanometers. There is a blue shift of the absorption and emission band for the main emission band when perovskite quantum dots are doped with palladium. And the most important conclusion is that the palladium dope fission lead to bromine quantum dots dope with palladium can be excited with a near-infrared laser, and they emit in the near-infrared as well. Thus, they could be used to stain the luminescent nanothermometers for applications such as hyperthermia or diagnosis of cancer. Thanks for your attention.